Hello everyone and welcome to the 5th lecture in the nanomedicine series, and today we're going to put together all of the other 4 lectures. We're going to finish up pharmacokinetics with barriers and routes of elimination, and then we're going to walk through the whole process from administration all the way through elimination. So let's first talk about the barriers. There's many different barriers, so after something is injected or ingested into the body, there's different barriers for it getting out of the bloodstream. So one of the barriers is just the bloodstream itself, the endothelium, so it has to get out of the bloodstream. And we've read a little bit about that in the doxal case, where there's different size holes in the blood vessels that make up the different size holes between the endothelium, which make up the blood vessels, and so bigger things like the liposomal particles typically do not go past that. But in disease states, for example, in tumors, the holes or fenestrations are much bigger, so bigger particles can pass through. So we're, in that case, taking advantage of biology and the fact that tumors have leaky vessels in order to preferentially get something bigger to accumulate there while being excluded from different tissues, so for example, the heart. Some of the barriers that we'll also talk about are the opsonization barriers, which are these little green things. The liver and the kidneys are routes of elimination. And then also if you need to get something inside a cell, so the plasma membrane becomes a barrier. So let's talk about all of the different barriers. So first, the endothelial barriers. Opsonization is one of them, and so opsonization, for good or bad, it just is, it's neither. But foreign particles will get covered with non-specific proteins. These are called opsonins. And the opsonins make them visible to the phagocytic cells, so those are the macrophages in the bloodstream, and then also the Kupffer cells, which are phagocytic in the liver. And then this makes it removed from circulation very quickly. So one of the ways that we protect against this is to put the PEG particles on it, the polyethylene glycol, and that makes them stealth, and so they're less likely to be covered with opsonins, and so therefore they'll stay in the bloodstream much longer. But if you want something to be in the bloodstream for a short amount of time, then the opsonization process is not bad. The other thing with the endothelium, and we'll talk about this in a little bit in more detail, is that healthy endothelium typically are very tight together and they have junctions, so only very small things can really get through these barriers, typically about 5 nanometers. So very small nanoparticles, the dendrimers, or small molecules can get through. However, larger particles, like for example the liposomes or some of the other particles, really do not pass through these barriers just because, or they don't pass through efficiently. Because they're too big to go through these junctions right here. However, in some physiologic or pathophysiologic cases, like with tumors or inflammation, the blood vessels have larger pores. And so small molecules can fit through, but also that opens them up. So fenestrations, these are the fenestrations, they are opened up for the nanoparticles to be able to go through and then deliver their active agent. In a tumor, this is called the enhanced permeability and retention effect. So that means that there's an enhanced permeability, but also the lymphatics of a tumor are disrupted, and so things don't get cleared from the tumor as quickly either. So things tend to accumulate more, and they stay around longer because they're not cleared. In the cases of, for example, what you've read for doxal, we're taking advantage of this natural biology or unnatural biology, since it's cancer, in order to preferentially change the biodistribution or the distribution of the particle in the body. So the EPR effect is a very important effect in being able to deliver something to a tumor. So here's what opsonins look like. This is the process of opsonization. So this is on the case where we're looking at bacterium, but if you can replace this with a nanoparticle, it doesn't matter. But an antibody or proteins in the blood can go ahead and bind to this nanoparticle or bacterium, activates the complement. So C3b is a protein that will then bind to this. Then on the phagocytes, you will get the antibody, this part, the Fc portion, binding to the Fc receptor, and C3b binding to its receptor CR1, which then activates phagocytosis, bringing inside the nanoparticle or the bacterium or virus or whatever it is inside the phagocytic cell, where these are then broken down. Unless you are trying to specifically target a macrophage, this means that you've removed some of your particle or some of your drug from the system, because that's what the phagocytic cells do, is they remove and sequester things. So again, if you're trying to hit macrophages, that's great. Making it non-PEG or non-stealth and just natural nanoparticles or naked nanoparticles, you will hit lots of macrophages. And even if you put PEG on them or you do things to try to target it away from macrophages, you'll still get a large amount of macrophage involvement. So this is what this looks like on an EM. So this is a nanoparticle right here, this Y. Here's the leading edge of a macrophage. And so look at this macrophage engulfing something that's actually just almost as big as itself. So this is really a nice and exciting image to show really how deformable these cells can be and how their membranes are not that real solid part. So another barrier is the mucosal barrier. And this is the barrier that's in the GI tract and also in the nose. So this is what a colon crypt looks like, and the colon crypts are the invaginations inside the colon, and most of the process of the colon is just to absorb the water that's left that hadn't been absorbed in the small intestine. So the colon doesn't absorb nutrients, it just absorbs water. To do this and to fit it inside the body, the colon has many, many invaginations, and these are crypts. So this is one crypt. Here's the nuclei in blue. Here's the cells, and then this red part is the lumen, so that's the opening. And inside this, to protect this colon, is a mucosal barrier. You also have mucus, obviously, in your nose and also in your lungs or your airway. So the mucus stops things from being able to get in and any kind of infection from happening. So let's talk about cell barriers. So barriers, these are barriers between cells. This happens in the endothelium and also when you're trying to get things past the mucosal barrier and into the lumen, past the lumen, past the mucosal barrier, and into, for example, the gut or other things. So one of these is a tight junction. So epithelium, let's go back to this. So epithelial are sheets of cells that provide the interface between masses of cells and a cavity or space. Except for the blood vessels are endothelium, but all of your organs have these spaces or lumens, and the epithelial are what line those lumen. The portion of the cell exposed to the lumen is called the apical surface, so that's up here. And the rest of the cell, the sides and the bottom, are the basolateral surface. Most epithelial cells are polar, with the nuclei most of the time in the basolateral surface and away from the apical surface. So tight junctions, they seal adjacent epithelial cells in a narrow band just beneath their apical surface. So here's the tight junctions. This tries to prevent things from being able to go through. They perform two vital functions. They prevent the passage of molecules and ions through the space between cells. So materials have to actually enter the cells by diffusion or active transport in order to pass through the tissue. This pathway provides control over what substances are allowed through. So only the things that have a way of transporting through the cell will actually be transported. They block the movement of integral membrane proteins, these red and green ovals, between the apical and basolateral surfaces, thus the special function of each surface. So again, that's the polarity. There's receptor-mediated endocytosis at the apical surface and exocytosis at the basolateral surface. And in this way, we can take advantage of this. If we know the biology and what's on the cell surface, we can get something, a nanoparticle, to be taken up by the cell, brought through the cell, and then secreted into the basolateral surface. Another type of junction is an adherence junction, and they provide the strong mechanical attachments between adjacent cells. They hold, for example, cardiac muscle cells tightly together as the heart expands and contracts. They hold the epithelial cells together. They're responsible for contact inhibition. So what that means is that when cells are touching each other and have these adherence junctions, they don't really proliferate. And that's good because we don't want them to proliferate because that's what forms a tumor. Some adherence junctions are present in narrow bands between connecting adjacent cells, and then others are present in discrete patches holding the cells together. So here's the adherence junction. There's cadherins that hold them together. They're on the cell surface, and then they bind catenins, and then also actin filaments, so they're responsible for the motility. These adherence junctions are built from cadherins. They are transmembrane proteins, so they are in the inside of the cell, go through the plasma membrane to the outside of the cell, and their extracellular segments bind together. Their intracellular segments bind to catenins, and catenins are connected to actin microfilaments. And one of the oncogenes that is frequently found in colon cancer, so oncogenes are genes that promote cancer, and that's beta-catenin. So it's the mutated version of beta-catenin, or APC, which is adenomyosis polyposis coli protein. Loss of functioning adherence junctions may also lead to tumor metastases. So it's very important for maintaining these tight barriers. Another junction or another barrier is a gap junction. So gap junctions are intercellular channels. They're about 1.5 to 2 nanometers in diameter. They permit the free passage between the cells of ions and small molecules, up to a molecular weight of about 1,000 Daltons. So again, when we're engineering something, if you want something to go through one of these junctions, you have to think about the size, so 1,000 Daltons. They're constructed from 4, sometimes 6 copies of one family of transmembrane proteins called connexins. And then because ions can flow through them, gap junctions permit changes in membrane potential to pass from cell to cell. So this is where the cardiac rhythm works. The action potential in the heart flows from cell to cell through the heart, providing the rhythmic contraction of the heartbeat. And the electrical synapses in the brain, the gap junctions will permit the arrival of an action potential at the synapses to be transmitted to the postsynaptic cell without the delay needed for the release of a neurotransmitter. So as the time of birth approaches, gap junctions between the smooth muscle cells of the uterus enable the coordinated, powerful contractions to begin. So again, these junctions are very important. Several inherited disorders of humans, such as congenital heart defects and congenital deafness, are caused by mutations in the proteins or mutations in the genes that encode connexins. Very important proteins. And then finally, the desmosomes. These are localized patches that hold 2 cells tightly together. They are common in epithelial, especially in the skin. Desmosomes are associated with intermediate filaments in the cytoplasm. Carcinomas are cancers of epithelia. However, the cells of carcinomas no longer have desmosomes, which we think maybe may account for their ability to metastasize. So here's this all put together, going through the some of the barriers. So this is an example of HIV. So here's cell-free HIV. It can bind to these cells and then through a transcytotic pathway, just pass from cell to cell. Here's a cell associated with HIV. For example, HIV can bind and be put into T cells. These cells can then deform and cause changes in the fenestrations of the epithelial or of the blood vessels. And the T cells themselves can transform so they can transmigrate through. These T cells can then bind to things like Langerhans cells or dendritic cells, and so go in there. Or they can cause, or in an ulcerated or an abraded situation, they can go direct entry through the breaches in the epithelial integrity. So there's different ways for things to be able to go through. Most of the time in the nanoparticle situation, this is going to be passive transport. They are going to get out through just the fenestration size. So anything that's bigger than 5 nanometers will not pass through healthy fenestrations, and so you're really looking at something that's an inflammatory response where the fenestrations are bigger to allow white blood cells to get through, and if a cell can get through, then of course a nanoparticle can get through. So most of those situations are really passive transport. There's very few situations where people have taken advantage of cell surface receptors in the endothelium in order to do this transcytosis section. So another barrier that's pretty interesting is the blood-brain barrier. So if you want to get anything into the brain, you really have to think about the blood-brain barrier. Now the blood-brain barrier, the brain is highly protected in the body. Most things do not reach the brain. We don't want things in the brain. The brain is really an important function. So this barrier has really, it is really exclusive for things. So the blood-brain barrier, or the BBB, results from the selectivity of the tight junctions. Again, we talked about tight junctions between the endothelial cells in the CNS, so central nervous system vessels. And these really restrict the passage of solutes. You can get glucose in there, but not much else. At the interface between the blood and the brain, endothelial cells and astrocytes are stitched together by structures called tight junctions. The tight junction is composed of smaller subunits, most of the time dimers, that are transmembrane proteins such as occludins, claudins, junctional adhesion molecule, ESAM, and others. So occludin, it's baked right in their name. This function is to stop things or prevent things from going into the blood, into the brain. Each of these transmembrane proteins is anchored into the endothelial cells by another protein complex that includes ZO1 and associated proteins. The blood-brain barrier is composed of high-density cells restricting passages of substances from the bloodstream much more than endothelial cells and capillaries elsewhere in the body. Astrocytes have cell projections called astrocytic feet. They surround the endothelial cells of the blood-brain barrier, here they are, right here, providing biochemical support to those cells. The blood-brain barrier is distinct from the similar blood cerebrospinal fluid barrier, a function of the choroidal cells of the choroid plexus, and from the blood-retinal barrier, which can be considered part of the whole. So these are very different. Several areas of the brain are not behind the blood-brain barrier, and one example is the pineal gland, which secretes the hormone melatonin directly into the systemic circulation. But for the most part, to get anything into the brain, if you need to treat a tumor in the brain or a seizure or a stroke or anything, any kind of pathology in the brain, you have to be careful and think about this blood-brain barrier. There are techniques to try to open it with radiofrequency ablation, and then people have also taken advantage of the fact that there are certain molecules on the blood-brain barrier, on the endothelium, that when you bind them, they can then do the transcytosis. And so in that way, that is that's an active process, it's active transport. So you'd need to decorate your nanoparticles with something that would allow the blood-brain barrier to take it up. But that's a barrier. For the most part, nanoparticles are excluded from the brain. So the pathophysiology, the blood-brain barrier acts very effectively to protect the brain from many common bacterial infections. So infections of the brain are very rare unless it's something like an amoeba or something, but those are still rare. So antibodies are too large to cross the blood-brain barrier. Infections of the brain, when they do occur, are very serious and difficult to treat. So that's the meningitis, that's the amoeba kind of thing. The blood-brain barrier becomes more permeable during inflammation, so some antibiotics can get across. Viruses easily bypass the blood-brain barrier by attaching themselves to circulating immune cells. So that could be a really interesting thing, is to potentially, and it hasn't been done, but to potentially mimic a virus to be able to drive your nanoparticle or drug into the brain. That could be a really interesting project. The blood-brain barrier is permeable to small and lipophilic molecules up to 800 AMU, but larger molecules are not transported unless, again, there's some active transport system available. This is one of the big stumbling blocks for drug delivery. An additional problem is the very effective drug efflux systems, which pump the drug back out of the cells. So the blood-brain barrier, it really does not like things to go across it. So drugs that target the brain, overcoming this difficulty of delivering therapeutic agents is a major challenge to treatment of most brain disorders. It has a neuroprotective role. The BBB functions to hinder the delivery of many potentially important diagnostic and therapeutic agents to the brain. Therapeutic molecules and genes that might otherwise be effective do not cross the blood-brain barrier in adequate amounts. So mechanisms for drug targeting in the brain involving going either through or behind the blood-brain barrier. So far things that have been used for this are disruption by osmotic means. So that's the radiofrequency ablation or osmosis. Biochemically, by the use of vasoactive substances like bradykinin, or even by localized exposure to high-intensity focus ultrasound or HIFU. Other strategies have been to use the endogenous transport systems, including carrier-mediated transporters such as glucose and amino acid carriers. Those wouldn't really work for a nanoparticle because they're too big. But what has worked is receptor-mediated transcytosis for insulin or transferrin. A lot of people have used transferrin and the transferrin receptor to get this through. And blocking of active efflux transporters such as P-glycoprotein. So strategies for drug delivery behind the blood-brain barrier. So the previous strategies are through. This is for behind, and that's usually surgery, so intracerebral implantation or convection-enhanced distribution. So nanoparticles have been used to try to help the transfer of drugs across the blood-brain barrier. Researchers have been trying to use liposomes loaded with different nanoparticles to gain access to the BBB. More research is needed, so this is really a hot area of research. People haven't really been able to do this, and I'm not aware of anything right now that's clinically available or even in clinical trials. So the potential for using blood-brain barrier opening to target specific agents to brain tumors is just beginning to be explored. So delivering drugs across the BBB is one of the most promising applications of nanotechnology in clinical neuroscience. Nanoparticles could potentially carry out multiple tasks in a predefined sequence, which is very important in the delivery of drugs across the blood-brain barrier. A significant amount of research in this area has been spent exploring methods of nanoparticle-mediated delivery of anti-neoplastic drugs. So these are drugs that are anti-tumor drugs to tumors in the central nervous system. For example, radio-labeled PEG, coated hexadecycloacrylate nanospheres, targeted and accumulated in a rat gliosarcoma. So again, this is in a rat. It's not ready to do in clinical trials due to the accumulation of the nanospheres in surrounding healthy tissue. It's also, we should note that vascular endothelial cells and pericytes are often abnormal in tumors, and the blood-brain barrier may not always be intact in brain tumors. However, by the time that the blood-brain barrier has broken down, it's really late for the tumor, and so we really need to find a way to get past the blood-brain barrier. Other factors such as astrocytes may contribute to the resistance of brain tumors to therapy. So the blood-brain barrier represents an important opportunity for students and for researchers to be able to really make a difference to delivering things to the brain. So this is an active area of research, a really exciting area of research. So let's talk about cell barriers. So you've got it past the, you got it out of the endothelium, and you've got it into the interstitium, and now you have to deliver your nanoparticle or your drug into the cell of interest. So the plasma membrane represents another barrier to this. Big things really just don't go through the plasma membrane. And so the principles, there's different principles of membrane transport. It's a selectively permeable barrier. It allows some things like ions, amino acids, to go through the plasma membrane. And protein-free lipid bilayers are highly impermeable to ions, and it's energetics of solute movement. So diffusion, there's three things. So diffusion is the spontaneous movement of material from a region of high concentration to low concentration. So if more things are on the outside, like sodium or potassium, then on the inside, it will just passively diffuse through. The free energy change during diffusion of non-electrolytes depends on the concentration gradient. And the free energy change during diffusion of electrolytes depends on the electrochemical gradient. So again, more things out here, less in there, it's going to go through there. Same thing, if something is highly accumulated inside the cell and there's less concentration on the outside, it will diffuse out. These are really specific though for ions and things. Most big things like a nanoparticle will not passively diffuse through a plasma membrane. So there's both active and passive transport. So solutes that can be transported, small nonpolar, example oxygen, large nonpolar, fatty acids, small polar water, they can all go through simple diffusion. They do not need facilitated diffusion, and they do not need active transport. They can just go back through. Things like large polar glucose or ions, sodium, potassium, calcium, they do not do simple diffusion. They will do facilitated diffusion and active transport. Thermodynamic properties, so direction relative to electrochemical gradient, they go down from that. Effect on entropy, that's increased or decreased. Metabolic energy required, no. For active transport, absolutely. You need energy to do that. Kinetic properties, carrier mediated, so that's things that are proteins that are endocytosed. No simple diffusion, yes, facilitated, and yes, these are active transport. Michaelis-Menten kinetics, so those are enzymes, or inhibition, yes, so, and they need energy. So uncharged molecules, if they're small enough, they can move down their concentration gradients directly across the lipid bilayer by simple diffusion. Most solutes can cross the membrane only if there's a membrane transport protein to transfer them. Passive transport in the same direction as a concentration gradient. Active transport is mediated by carrier proteins against a concentration gradient that require an input of energy. So this is endocytosis. We take advantage of this a lot when we're talking about targeted imaging or targeted therapy. And we will talk about that later in the class. So here's some examples of passive and active transport. These are two classes of membrane proteins. They're carrier proteins. Here's a channel protein, and so this can just go through. Passive transport, there's no conformation change. It can just go through. But carrier-mediated or facilitated diffusion is this molecule comes in, it binds to this receptor, and in the process of binding that receptor, it changes the conformation of it, and then it allows it to move through the membrane. And then this is active transport, where it's going against the concentration gradient. So it will go in, bind, and then need energy to be released. So there's tens of different types of ion channels. They all share distinctive characteristics. They are formed of glycoproteins. They're proteins with sugar molecules attached, and they traverse the cell membrane. So they're transmembrane. They have an extracellular part, a membrane part, and an intracellular part. They are formed by aggregation of subunits. Each is a protein by itself, and they form a cylindrical configuration that must have a pore. So they form a kind of tube that are in the membrane. These subunits can be copies of the same protein, or they're a combination of different proteins which are it's called a heteroprotein. In some cases, the subunits are formed by the folding of some very large but simple proteins. Ions will cross through their channels by electrical potential or concentration differences across the cell membrane. Kind of in the simplest example is the electron transport chain in the ATP synthase. The passage itself does not require metabolic energy expenditure. The energy derives from the chemical forces of diffusion, osmosis, and electrochemical equilibrium. There are other types of membrane-spanning proteins that move ions against these forces, but it costs a lot of energy. They are not considered channels, and they are called ion pumps. These channels are selected of the types of ions it allows to cross. So in this sense, ion channels are not only open holes of different diameter, since channels that allow the passage of a certain ion do not allow passage of larger or even smaller ones. The type of ion that is allowed to cross depends on the electrochemical configuration of the protein subunits, especially of the side that faces inside the pore. So for example, something that will pass calcium will not pass potassium or will not allow sodium to go through. So it's very specific for each type of ion. It is common that a type of ion channel will allow several kinds of ions to cross, especially if they all share the same charge. The variety and number of ion channels that a cell has is determined by its type. Its distribution can be used to determine the type of neuron or other cell that is being measured. Additionally, the distribution within the same cell can have great variability. For instance, the axon of neurons have a larger density of sodium and potassium channels than the rest of the cell body. So there are different mechanisms for opening or closing of an ion channel. It can be phosphorylated and dephosphorylated, or the protein structural changes can be achieved. The coupling of a messenger, typically this is the case for neurotransmitters, causes it to open. These are called direct gated receptors or ionotropic receptors here. So phosphorylation can cause a conformational change that can open it. Here's where a neurotransmitter binds, which can cause this to open. And then also a change in membrane potential can cause a conformational change. These are voltage sensitive channels. Those are more often in the heart. So here's a different example, and this is more appropriate for what we're talking about for nanoparticles. This is phagocytosis. This is the uptake of larger particles, macromolecules, cell debris, even microorganisms in other cells. It's usually restricted to specialized cells called phagocytes. Those are macrophages. It's initiated by cellular contact or nanoparticle contact with an appropriate target, and it can be stimulated by obstinance, especially in the Kupffer cells and in the macrophages. For tumor cells have more active membranes, and so they are more likely to take up nanoparticles than healthy cells. Another example is receptor-mediated endocytosis. So phagocytosis is nonspecific. That's just the cell sampling its outside domain once something binds. But this is really specific, and this will come into play when we start really talking about targeting. We take advantage of receptor-mediated endocytosis quite frequently. And so what happens is there's a receptor on the cell surface, a ligand or a nanoparticle with a ligand that's specific for this receptor will come and bind. There will be some lateral diffusion, and then from the lateral diffusion there will start to be a coated pit. And there are adapter proteins and clathrin, this is one example, clathrin, and dynamin. And so clathrin will bind, and so will the adapter proteins, and form this clathrin coated pit. And dynamin is a membrane protein that allows the use of energy, or a motor protein that allows this clathrin coated pit to pinch off from the membrane and to close off and become a vesicle. So the vesicle forms, it's a coated vesicle. It then gets uncoated, so clathrin is then removed. It fuses with the early endosome and it releases the ligand. It can be then transported to late endosome for digestion, and then from there the receptors are transported back to the cell surface. Things can be transcytosed, so if you want your nanoparticles to stay intact and be transcytosed, through, for example, a blood-brain barrier or through the endothelium. That can happen. Or you can make your nanoparticles degradable, so pH degradable, so that way they will degrade inside these endosomes and lysosomes, which are low pH, and then they can release the small molecule into the cell, and so that way you can treat that cell with a small molecule. So it depends on where you want to deliver it. And we take advantage of this, like I said, all the time, and I'll show you some examples later on when we start talking about targeting. So those are all of the barriers and different strategies of getting around them. And so now we're to the last part of the distribution. So we did the routes of administration. After the drug or the nanoparticle gets inside the body, we have modeled mathematically the different organs and where everything goes. And everything, though, in the final step, everything that gets put into the body has to be eliminated somehow. And there's three main routes of elimination: the liver or the reticuloendothelial system, the kidneys and the urine, or out through the lungs. There is a fourth one, the skin and through sweat, but it accounts for such a small amount of the elimination that we're really not going to focus on it. The liver is the main system. The reticuloendothelial system is the main system for nanoparticles. Anything larger than 32,000 daltons or 32 kilodaltons cannot be filtered through the kidneys and have to be eliminated somewhere else. That's typically by the phagocytic cells in the lymph node, the spleen, and also the liver. So that's the reticuloendothelial system. So no matter what, you cannot eliminate the liver and the spleen from taking up your nanoparticles. It's always going to be there. So here's our organs, here's the spleen, here's the liver, and then here's lymph nodes throughout the body. Anywhere where a macrophage or a phagocytic cell will reside, that's the reticuloendothelial system. The liver is very important. It's the detoxification organ. So toxins will come in step 1, step 2, and then step 3 are the waste products. They become water soluble, and they're eliminated from the body by the gallbladder, bile, and into the bowel. So that's the feces, or from the kidneys and the urine. So once they're processed, they can still go through the kidneys and the urine. They're just broken up into something very much smaller. So the toxin lists are metabolic end products, microorganisms, nanoparticles, contaminants, pollutants, insecticides, pesticides, food additives, drugs, alcohol, you name it. The liver is a very, very important function. Here's the required nutrients for step 1. The B vitamins, folic acid, glutathione, antioxidants, carotenoids. So those are the beta carotene things in carrots. Vitamin E, vitamin C. For step 2, required nutrients, amino acids, glutamine, glycine, taurine, cysteine, sulfonated phytochemicals found in garlic and cruciferous vegetables. So really good. But the liver is really important. And inside the liver are the Kupffer cells. And the Kupffer cells are the phagocytic cells, and they are what are going to take up your nanoparticle and eliminate them from the body. And this isn't all bad. It becomes an engineering problem. Getting enough nanoparticle into the tumor or into the pathology or the disease state that it's efficacious, but you also have to have these things eliminated from the body, so the liver has to be able to take up some of these and eliminate them. So you just don't want to do that first. You want it to hit the tumor first. And then here from the liver, things get broken up, and then it's eliminated through the large intestine or the colon. So again, here's the ascending colon, here's the transverse colon, and then here's the descending colon. And then again, here are those crypts. So you have a lot of surface area lumen for absorbing water, which is really important, and then passing through the feces and eliminating things that are not good. So that's the reticuloendothelial system. Again, very important. That is the main route for a nanoparticle. One of the main routes and where we lose a lot of the nanoparticle. The kidneys and the urine are the next. So anything under 32 kilodaltons, so smaller things, dendrimers, small molecules, primarily will be eliminated through the urine. They can be filtered out by the kidneys. Here's the subsection of the kidneys. Here's your cortex. They can be filtered out through the nephrons in here, and then put in through the urine, so no problem. Things that are larger, the 32 kilodaltons, will go through the reticuloendothelial system. They'll be broken down by the Kupffer cells in the liver, and then they can still be put back in, the smaller pieces can be put back into the bloodstream and then filtered out through the kidneys. And the kidneys filter it, goes through the blood in the kidneys to the bladder, and then out through the urine. So again, this is through the smaller subunits, the nephrons. There's about 1 million nephrons in each kidney. They filter water and substances out of the blood to produce urine. So there's the nephron. And inside that nephron is a glomerulus. So here's the glomeruli and the Bowman's capsule. So this is where all of the exchange takes place. And then finally, the last system is the lung system. We've already talked about the lungs when we're doing inhalation. Well, any substance that can be volatilized can be exhaled through the lungs. And so, again, anything that but it has to be able to be volatilized. It has to be able to turn from a liquid in the bloodstream into a gas out of the lungs. So there's mechanics of breathing. The rib cage, when you inhale, the rib cage moves up and out. The diaphragm moves down. The lungs expand. When you exhale, the rib cage moves down and in. Lungs get smaller. Diaphragm moves up, and then the air leaves. And this is all due to CO2 concentration. So the more carbon dioxide you have, the more you have to breathe. Inside here are the alveoli, so these are openings inside the lungs, and this is where the gas exchange occurs. So where the green and the blue arrows are, that's the alveoli. There's endothelial running through it, and that's exactly where that gas exchange occurs. The alveoli are very small, and so when you have lung tumors or you have bronchitis or something that will obstruct your alveoli, you cannot exchange oxygen and carbon dioxide, and so that hampers your ability to breathe and get oxygen inside the body. So putting it all together, we've talked through the various lectures about the administration. We've talked about the distribution, and the distribution in the 1-compartment model, 2-compartment model, 3-compartment model, and then also taking into account things with absorption. So if it's not an intravenous route of administration, if it's other things, talking about how that changes the distribution and how things will accumulate in the different organs. I haven't really talked about the PBPK model, so those are the physiological based pharmacokinetic models. We are going to do that in the worksheet that we're going to do on Monday or Wednesday. Then we've just talked about elimination. So although we've had the K elimination curve or constants or rates, we've talked about elimination. It's still important to know about the roots of elimination just because we want to sometimes modify around it. For example, if you have a liver tumor and you want to be able to image a liver tumor, you don't want something that's over 32 kilodaltons because that would be taken up by the liver, the Kupffer cells in the liver, and so your background would be really high. You wouldn't be able to see the tumor because you'd have so much in the normal, healthy parts of the liver. Likewise, if you wanted to see something in the kidney, you'd want to make sure that you have it so it clears very quickly from the bloodstream, so make it smaller, but it would still accumulate in the tumor. So again, you're playing with clearance here, you're playing with blood half-lives, and you're thinking about exactly how we are engineering things to overcome or to use some of the body's natural barriers or just natural processes in it. You just have to be aware of all those things. So administration, distribution, and elimination. And the worksheet that we will do, we'll put it all together. So thank you for listening to Nanomedicine 5, and I look forward to seeing you in class.